My name is Dr. Bruce Baldwin, and I'm the department head for, I think I'll say, chemistry biology. I know that more than half my department is now offended, so, all right. But anyway, I'm the head guy. So, this is our newest member of the chemistry biology department. This is Dr. Aaron Wyman. He is making our lives much, much more easy to live by being who he is and taking classes and stuff. I mean, teaching them, not taking them himself. Today he's going to talk about stuff that he's done in the past with students, I understand.
Weird.
Not just all by yourself in a little tiny room. No. You're affecting the lives of students, and we hope that you'll be willing to do the same thing with some of the students out here. Maybe students that we don't yet know about, Dr. Wyman.
Totally possible.
Because they haven't stepped forward and said, I want to do some research that has to do with biomineralization. Because that sounds really cool. Sounds cool even to me, and I'm a chemist, not a biologist. So that's saying a lot. Please take it away.
Thank you, sir. Thanks, Bruce. And look, you all came here voluntarily. Fascinating. No? Oh, I'm hurt. 6 months ago, I stood up here and I put on the best show I could, and I am truly grateful to be here. My wife is grateful to be here. My child, he's grateful to be here. He doesn't know it, but he is. He's not quite old enough to realize that. I couldn't be happier to be here, and I hope to know all of you within the next month to 2 months, maybe 4 months. My office is 108C. Come by. Let's talk. It's okay. I might even have candy. I may not share it, but I might have candy. I'm going to talk about biomineralization today, which is important to all of us. Okay? And as part of this concept, I have what I call the big plan. I got this idea in my head. It was one of two ideas I had, along with proposing to my wife on my own. This idea of how could I get you all of you undergrads, engaged in research activities. I needed a mechanism for that. I want you to become critical thinkers. I don't necessarily care if you become researchers, because then you're competition. I don't really want you to come get my job just yet. Wait 35 years, then we'll talk. But I need you to think critically. I need you to think about how to solve problems. And so I needed a research platform that was amenable to you, that had your interest, that you could do. You're not necessarily ready to splice embryonic DNA. Maybe you are, but most likely not. Okay, so we're going to think a little bit more broad. It has to be cost effective, preferably close to zero, and time effective. Okay? And we have to have the machinery, the equipment, and the capacity for maintenance. When I interviewed here, I saw all of those things. In fact, they basically screamed at me. Like, oh, this is nice. I want you all to have active hands in novel, new, independent research capacities as frequently as possible. I want you to learn how to make pancakes on your own. Trust me, you haven't lived until you've made pancakes by yourself. Trust me, all right? I based these ideas off of what I had as an undergrad myself and efforts I saw for single classes, but I want to think more broadly. How could I tie my research into everything I did? Everything. And so, in doing so, I want you students to understand, and this isn't totally your fault. I blame you for a lot of things. Your inability to write because you text all the time, for example. I blame you for that. The reality is, though, many of you think of education as a discrete set of shoeboxes. All right, I fulfilled my organic chem shoebox. I have now fulfilled my genetics shoebox. I tried to fill my organic chem shoebox. I tried to fill my genetics box. Education, learning is not filling boxes. It's spilling all the boxes on the floor and kicking them around and looking for a clean pair of underwear. That is when you know something. Some of you are laughing. Does that mean underwear is not? Oh, okay, no. We'll move past that. Now we're uncomfortable. At previous institutions, I combined these ideas in classes I were teaching. And I'm taking the same approach here. So if you take my botany class, actually many of you will, congratulations, you'll be engaging in independent work. You don't necessarily know it off the bat. I'll tell you eventually. If you're in genetics, you're full-fledged deep in it now. You may or may not be happy about this. I recommend you are, at least until the end of the semester. All right? And in micro, I promise, we're going to do the same thing. You're going to engage in novel ideas. I'm going to have you apply what you learn to a novel idea. If we make headway, you're going to be on a manuscript. And is that useful to you? Shake your heads yes, so I can hear the rattle. All right. Biomineralization involves the biological control of formation of insoluble solids. Oh, that's a fancy way of saying. All right. It typically involves calcium, either calcium phosphate, calcium oxalate, bench. Has roles in support, defense, feeding, maintaining ion concentrations. Oh, well, that sounds kind of fundamental. Yeah. Here's an example. Those are called bones and teeth. Ooh, he's smart. Yeah. All right. All right. Ironically, this is a cast mold. It's plastic. None of that's real. Now, calcium hydroxyapatite is what makes up your bones and your teeth. But keep in mind, those don't form by accident. Imagine if all of your bones formed as amorphous masses. Would you look like you? No, it'd be kind of a lump, a rocky lump. That's not fun. I want to have bones. I want to have teeth. I want to be able to move. So we have to have proteins and carbohydrates to help guide formation of that calcium solid. Right? This is non-random. These are sea anemones, sea urchins. They contain calcium carbonate, a calcite, and they have proteins and carbohydrates responsible for making those long, hard tines. These are crystals formed in different plant species. And if you've had the joy of ever hoeing velvet leaf, and to be honest, you haven't actually hoed velvet leaf, you know what I'm talking about, you pull it out with your bare hands. And I've spent many an unhappy day doing that, which is why I'm here now and not farming with my father. Because farming is work. Teaching you all? No. Pleasure. All right. Crystals form in different species of different morphologies, numbers, sizes, and they all involve calcium oxalate and its effective, specific proteins and carbohydrates. And I got excited when I saw this idea. I'm like, hey, students can work with this because you all like teeth and bones. You've told me so. You want to become a dentist or a doctor. You all like the idea of eating food. That comes from plants. Oh, yeah, it does. A good point. Plants have crystals. I've got a hook now. All right. What's nice about this process is it involves basic fundamental science. How are crystals formed? What controls their nucleation? What controls the morphology they have? It involves defined biological factors, which are gene-controlled. And remind yourself, you have genes, and so do plants. Many MDs don't know that. That's a scary thing you've come to learn. Your MD, always be nice to them. They have needles. But understand that many of them aren't necessarily the best and brightest. They've finished in their class. They got the degree. They're nice people. Don't misunderstand me. But problem solving may not be their highest strength. One of the interesting things is that there are common mechanisms between organisms. Life replicates itself. It continues. So if we can study a mechanism in one species, we might be able to apply it to another. Ooh, hey. All right. For those of you who are interested in building stuff, I predict that by the time you all are 50, we're going to be able to rebuild bone on the fly. If you're missing a bone, if you have a shattered bone, we're going to rebuild that bone on the fly to an exact size and shape in a biological fashion. Wouldn't that be kind of cool? And we can replace teeth. My grandfather still says he'd like to have some of those teeth back. I agree with him. Just keep that in mind. Okay? We can use what we learned from studying these crystals, how these crystals are formed, to make artificial inorganic solids, right? Like bones and teeth. And to purify the earth, because it turns out humans aren't really good about cleaning up after themselves. You have a roommate perhaps like this, or maybe you are that roommate and you just don't want to admit it. We aren't very good about cleaning up ourselves. We are looking at how can we use the formation of crystals to clean the earth, to restore it to what God gave us. Oh, that's interesting. And then there's these. Now, people in my genetics class know what this is because they've seen this picture before. These came out a very small opening. Ow. Under force. Ah, yeah, very good. Oh, it's the correct answer right now. Okay. Yeah, these are called kidney stones. They started in you, and then they came out of you. Those are kind of nice because they're smooth. I've seen some that aren't. 1 out of 8 of you gentlemen will have one. Look around, gentlemen. 1 8. Ladies, you're at 1 out of 30. So here's the trade-off. We let you do the birthing thing because we are not strong enough for that. We'll pass kidney stones a few more times. That's going to be a trade-off. This is an otter from the Smithsonian Zoo. Those are stones. They're almost the size of the poor otter's kidneys. Think the otter's in trouble? Yeah. Okay. It turns out, yeah, there are proteins directly involved and carbohydrates directly involved in forming calcium oxalate stones. Here's the problem with kidney stones. They don't kill you. Oh, they hurt. They cause infection, but they don't kill you. So how much funding do you think they get? We're trying to change that. Because here's reality. 1 out of 8 guys is going to have this. 1 out of 30 ladies are going to have this. We need to push on this. We need to push on this. That's the last time we saw him. It was weird. All right. I like plants. I like plants because they have never bitten me. They don't fly. They don't smell bad. Bless PETA, but there is no PETA for the ethical treatment of plants. I can grind them up. No one cares. It's cool. All right? And they give me fruit. Oh, that's a good sign. So here's my friend skunk grape. Skunk grape makes these beautiful sets of crystals that glow under polarized light.
Oh, yeah.
Yeah. That's part of chemistry. Dr. Coulson will be happy to show you part of that. Okay. And what's nice about these systems is they have direct application to basic sciences, to applied sciences, bioremediation, and biomedical research. This topic I saw is perfect for undergrads because this fits at least one category for all of you. If you don't like any of these things, you're probably not interested in the sciences, and that's okay. God makes us all different, gives us all unique perspectives. I'm going to stop right there and continue. Plant families make different types of crystals, shape, size, number, form. So we can use them for classification purposes. Frequently they're calcium oxalate, but they could be calcium phosphate, they could be glass. What's great is that they're easy to study by the eye. We can use microscopes, cytology, and we have molecular and genetic tools we've worked out to study them. We're not working blind. Okay? What's nice about them is for a plant, they help protect you against cows. Imagine chewing on leaves that are filled with hypodermic needles. That describes my breakfast. Wait a minute. Right? The reality is that's going to hurt. Okay? They also help the plant regulate calcium. Last you checked, did plants move very much? Now think about that. Unless it's an ant, it's not going very far. And we're grateful for that. Thank you. Thank you very much. All right? They have to capture calcium and maintain it. Calcium is absolutely essential for biological function, be you a plant, be you a bunny, be you. Okay? All right. Grape former are called Raphides. These are long, needle-shaped crystals, or druses. These are these mace-like heads. Okay. And they form in what are called idioblasts. These are specialized cells. You find them throughout the plant. And they form in a bag. Okay. That bag contains a number of acids, carbohydrates, and proteins that seem to be directly involved in controlling how crystals form. But we really have no idea what exactly is in that bag. We have a guess, but we don't know. So it's, I'm one of 6 in the world working on it, so my competition is not fierce. I'm also the most handsome, so I automatically win. Around each crystal is a membrane we call a crystal chamber. That's the outside of that bag, right? That helps define the shape and size of that crystal. Right? But we have no idea what defines the size exactly, the morphology, the direction of growth. We have ideas, but we don't have good concrete evidence yet. Cool. Right? But we do have animal-based systems. We have a pretty good idea how egg shells form. We have a pretty good idea how bones form. And so we've tried to apply what we've learned from animal systems back to the plants. Okay? Animal systems, like plant systems, have these biological membranes, oftentimes surrounding the growing bone or the growing teeth. They have matrices. They have these compilations of acids and bases and sugars and proteins that seem to be involved. And so I decided to focus on proteins because proteins are encoded by these funny things called genes. And genes can be found, manipulated, and studied. Right? And you've done so outside of an organism. And in doing so, my hope is that those proteins will lead me to answers on basic questions, on biomimetics. I have a collaborator at Stanford who is, every time I see him, which is every 6 months, reminds me that I need to send him the next round of clones because he works in a materials science lab. Not one person in his lab understands the concept of a cell, but he's fascinated with the idea that biologically we could control like the size and shape of a crystal. And remember, those crystals are in a wet environment. Do you think there's a value to making hard substances under controlled conditions in a wet environment? Think underground cement, underwater cement, folks. There's an advantage. All right? And of course, kidney stones, which hurt. I don't really need to explain that anymore, I don't think. So my lab's looking at what are called RAPs. No, I didn't come up with that one, but we're going to have some fun with that. These are Raphide associating proteins. They're proteins that either help stimulate or inhibit the growth of a crystal. All right. And there's different layers of RAPs. There's RAPs that are physically in the membrane itself, so they're on the outside of that crystal. And then there are proteins that you can find just within the crystal itself. And then there's crystals that act as seeds on the inside. We know they're there. We can see them by gel, but we don't know what they are still. We're trying to figure that out. So here are my first 3 rap artists, and I'm not apologizing. This was JP's idea. I'm going to throw him under the bus. All right. Austin has finished up IU Medical School. He's still debating which residency program he's going to go into. Must be nice to have options. All right. JP is a resident in orthopedic surgery in Chicago. I am ashamed to say I do not know what happened to Davida. I have not heard from him since 2008, and I've tried. I'm hopeful he's happy. I'm hopeful he is content. These 3 gentlemen worked really hard with me. Okay? We worked together. We collaborated. Research is collaborative. I'm not going to sit there and tell you a list of things to do. I'm going to make suggestions, and sometimes the suggestions become, you will do this. But I'd rather work with you, not through you. There's a big difference between those 2 things. We would take grape leaves, we'd freeze them in liquid nitrogen. Ooh, yeah, but not like Councilman. It's not quite as exciting what Dr. Councilman does. So don't get too thrilled. We're not blowing anything up. We're not making clouds. Freeze the tissue down, grind it down, and physically extract, sift out crystals. Okay? This takes time. You have these things called iPods, I believe, or iPhones, or somethings you can listen to music on. Yeah, the gentleman used to do this for hours. They just sift and listen to horrifying music. Youth, I don't understand any of you. We would take those crystals and we would solubilize them in different detergents under heat to extract proteins from the crystal. And we'd run the crystal on a gel, and the various bands you see, those are theoretically individual proteins. We take razor blades and cut them free. We digest them with the trypsin enzyme, and we send them off to what's called a multi-time-of-flight mass spectrometer, which is a fancy way of saying weight scale. Ooh. Again, Dr. Council will be happy to show you exactly how this works. Just ask him. All right? And if he sends you back to me, I'll send you back to him. It'll be fun for a while. All right. All right. MALDI time-of-flight MS allows you to get a literal fingerprint of a protein. It allows you to determine a mass to an exceptionally accurate value. So much so that you can take the mass and back calculate to what the amino acid sequence had to be. It's like taking, you live with a group of 5 people, it's like taking a scale, putting the person on the scale, and knowing exactly who took the last donut. Of course, you may already know from the crumbs. Come on, dude, that wasn't me. Dude, crumbs all over. Okay. From that amino acid sequence, we can go into online libraries, okay, at NCBI or using a mascot search engine, and we can look for proteins that are known to have that sequence. And in doing so, we can make a pretty good guess that the protein we pulled out of the crystal is similar, is a homolog at least, to the one in the crystal. You can say, okay, yeah, that probably is that protein. And these are data. The higher the number, the better the fit. These were preliminary data. We've actually gotten into the hundreds now. Those are pretty good hits. We've pulled out chaperones, transporters, signaling molecules. Ironically, calcium interacting proteins. Let's see, these are made of calcium. That makes sense. Yeah, that's a pretty logical thing. Cytoskeletal elements. Now, what makes me excited are the proteins of unknown function. That means that we don't know what they are. They could be really cool. I like really cool. I, unfortunately, now have around 65 targets. And there's how many of me? Oh, yes, one. Big ego, but still only one. Problem. Ooh, hey. Hey.
There are 85 students.
There are a lot of bodies in this room. You know what I'm thinking, don't you? My genetics class is going, oh, no, he started thinking again. Why does he do that? I need you. Okay? It also says, I need you. I need you to help me with this. Now, am I going to force you? If you're in my genetics class, yes. If you're in my botany class, probably. But if you're interested, if I have your attention, come talk to me. I don't bite. Now. All right. What I've done, and what students have done with me, is we design primers to amplify plant-based gene sequences that encode for copies of these proteins. All right? We clone them out, and we can express those genes in either bacteria or yeast to get functioning protein. We've started doing this. And we can play with the protein then. For example, we can ask ourselves, does the protein induce crystal formation, or does it induce crystals to break up? In our inorganic chemistry, you're going to work with UV-vis spectrophotometers. It's a very cool way of watching solutions get cloudy or clear. Cloudy or clear. Very simple test. Right? I have a collaborator, Mary Alice Webb at Purdue, who has access to an electron microscope. Exceptionally high-end resolution, going down into individual crystal molecules at extreme levels. We can look at, for example, do the crystals change shape upon being exposed to this protein? Do they change form? Do they change in number? Right. We can also see, do these proteins interact directly with the crystal? We have all kinds of tools we can work with. And we don't have to do it here. Remember, we're part of a larger community. We want you to critically participate. Oh, come on. And they can say, very well, thank you. Right? One of you gets a diploma. The rest of you I'm getting worried about. All right. All right. We need you to go out. But we're going to send you out as Christian scientists. That's our goal. Or as Christian professionals. Or at the very least, Christian citizens who are employed. Okay? All right? Keep that in mind. But I need you to be ready to go out, to work with everyone else. All right? We can't do it all by ourselves here. Trust me, we can't. This is one of my shining stars. She's about this tall. Kristen, you may have her beat. Right? This is Jessica. Jessica has just started a PhD program in biomedical sciences. She had 12 offers. I didn't have 12 offers. I had significantly fewer than that. She came to me. She said, Dr. Wyman, I have a problem. I go, yeah. She said, they all said yes. I told you they were going to. She said, what do I do? I go, well, you think about it. We talk about some more. So she went on 12 interviews. You don't want to do 12 interviews in a month and a half, by the way. You miss a lot of class, it turns out. She decided to go to University of Alabama, Birmingham, because they gave her $30,000 a year. yeah, that's what I said. Really? Yeah. And a housing allowance. Oh. You have a personal servant by chance? No. Okay, fine. Like an attaché or a posse of some kind? No. All right. She was looking at nephrocalcin. Nephrocalcin is found in mammals. It has a defined impact on crystal formation in mammals. Right? When it's mutated in mammals, it will form, kidney stones increase, the prevalence increases. She looked at, can we find a homolog in plants? And she found one. And she was in the process of cloning it out when she had the audacity to graduate. She said she had to leave. Ironically, so did I. So we both were okay with that. I introduced this kind of work, though, both at an individual level and at class level. I had entire classes cloning out these genes. I had entire classes working to express and purify the proteins. Right? In my classes, with me, as researchers, you are going to engage in novel research. You're going to learn the skills you need And it's not just me. It's true of all of my colleagues. The difference is I'm talking today. Okay? So I don't make it sound like I'm the only one doing this. Trust me, all my colleagues are doing this. You've seen it. You know. We are trying to get you involved actively in this field. I could teach you how to make an omelet, but it's more fun, frankly, to watch you make one. You have to botch science. You have to botch it. Not losing a hand. Yeah. Those are different things. Not losing your partner's hand. But you have to botch science to master it, to understand it, to command it. Oh, man, I don't like failure. Trust me, this is the wrong field if you don't like failure. Well, or at least accept failure. All right. This led to another set of questions. All right, fine. We're looking at what do these genes do outside of an organism, but what do they do in an organism? We need an in vivo model. Oh, okay, fine, I can do that. Grape are lousy for genetics. They grow slowly. Your students eat the seeds and the fruits. Yeah, they require care. Oh, mustard. Yeah, nobody cares about mustard. Good. Nobody wants to do it. Raphides thaliana is the fruit fly of the plant world. It's small, it grows fast. My genetics students right now are shaking uncontrollably. They've been growing for 6 weeks and they don't want to see them anymore. And what's nice about Raphides is it has homologs, copies of the genes found both in mammals and in grape. So we can use it as a model. Cool. All right. And our question is, if we mess with the expression of those genes, what happens to the plant? Oh, that's an interesting question. You don't really want to do that to dogs, it turns out. People get upset when you do that to dogs or rats, and especially people. But plants, yeah, that's fine. Go ahead. This is Cheryl. Cheryl has finished up her master's degree in botany plant path. She's still working on getting into genetic counseling graduate programs. I think she's going to get in this year. I really do. She's been really working at it. Cheryl was an absolute phenomenal component of my work. She's holding Raphides. It's a cute little flowering plant. You can play with it. You can modify it. You can add mutations. You can cut down gene expression. It grows quickly. It fits in a small space, and it will form biominerals if we make it. So it's a cute little thing, and it doesn't walk around. Nor does it fly, and doesn't smell bad. So these are all positives. We're taking a reverse genetic approach. We're saying, okay, there's a gene. What happens if I botch its expression? I'm going to intentionally mess with its expression and see what happens. All right. The example I typically use in my genetics classes, and this seems to work well with you all. You receive a text from your roommate. Hey, I'm at the mall. You come get me. Okay. But what if that message was, hey, I'm at, is that a problem for you? Perhaps not. Perhaps you don't care. All right. The Christian attitude is, I do care, and now I'm worried. That would be the correct answer. You really don't know what's going on. You have a truncated message. That's what we're doing here. We're going to add artificial DNA sequences into our genes and say, what happens to its expression? We're going to eliminate the large amount of that message. Right. These mutations have been made for us. Sadly enough, it was at that school south of us. It's in Columbus, which will not be spoken of ever by me, by name. I have one school, it's 45 minutes east of here, and it has a cool theme song you may have heard, and neat winged helmets. No bias, no bias. All right. That institution has been kind enough to generate these Arabidopsis mutant lines and make them available at $7 a pop. Sweet. All right. These are two of my former genetics classes. All right. Emily here's on the left. She's going to be going to vet school in the next year. Elisa, funny enough, is going to be a teacher. Jim was working for the Illinois DNR, and Bianca's on her way to a master's program in genetics, not genetics, nursing, sorry. They all worked with a very similar setup you all will get the chance to play with in genetics. Right? And among the genes they played with, and I've added some since that point, are a huge number that have been shown to be involved in forming human kidney stones. So all of these genes lead to proteins that end up in kidney stones. And the cool thing is, Arabidopsis has homologs to all of them. And we have no idea what those genes do in Arabidopsis. Which means if you can figure it out, you can get a big star on your resume. I have your attention now, I do believe. Or maybe some of you are just nodding back and forth, and that's okay too. All right. This is Sam. Sam has finished up her degree. She is actively looking for a teaching job right now. If you are an education major, come talk to me. We can commiserate. No. Come. Few of you got that. Good. I have lots of projects that you can take with you to your future job. Do you think it would be cool as an education major to walk in and tell your future employers, potential employers, I'm going to have my students doing novel research at this cost? Would you possibly get your future employer's attention? So there's no one in here who's going to be teaching. This is a bad sign for us. All right. Well, the answer is yes, you're going to get their attention. Come talk to me. We can set you up with projects that will help make you a more attractive job candidate and will help your students achieve more. And if your students achieve more, everyone wins. All right? Now, is there any point to all of this? Have you actually gotten anywhere? I was part of a research team along with Pri. Pri is now back in Thailand. She's her home country. She's about 2 hours from her parents. Her parents are both PhDs at a different institution, and her husband's at the same school she is. Phenomenal graduate student, extremely nice, lots of fun to work with. Jessica and I had so much fun that she is now finishing her Master's of Divinity. I actually brought her closer to God. That's how we both are saying it. We talk every 2-3 months by email. She's a sweet girl. She had always, when she was actually in the lab as a summer student, she said flat out, she said, I truly love science, but God is talking to me on a daily basis. And I said, if God's talking to you on a daily basis, sweetie, you need to follow that path. Because as much fun as I am, I'm really not the way to go. And she's like, yeah, I agree. And so she ended up finishing off a biology/theology degree at Calvin, and is now finishing her master's. And we have Cheryl. We worked on a gene actively involved in maintaining grape crystals. And the paper is done. And one of the things you'll learn is that bosses test you. And I'm being still tested because that paper is still on that desk, and it's been there for 4 years. But at some point, we're going to release that paper. We actually have made progress enough to have one good publication. It just hasn't gotten out yet. All right. This was my molecular class last year. Again, I want to enforce. In my classes, you are going to engage in biomineralization research. And I will tell you are. It won't be a surprise. Like, ha-ha. All right. I integrate, I use my research component to help give you the skills you need for future work. Right? For example, either cloning out individual plant genes, or expressing those genes, or looking at what happens in bacteria. There are a large number of bacteria that form calcium oxalate. In fact, there are a number of genus that we find in mammalian systems. If you lack them as part of your microfauna, you form kidney stones more often. That's kind of cool. I haven't had a chance to tell you those. I'm going to now. Cool. All right. I'm a big proponent of cleaning the earth. We made it a mess. We sort of owe it to ourselves, kind of. Yeah, we do. All right. So one of the projects I haven't pulled it out recently, Jessica and Carrie, who's now a medical student at IU, we worked at, could we use crystals to capture heavy metals like strontium or barium or selenium? The answer is yes, we can. They're really good at it. One of the cool things about plants have these things called vacuoles. You may have heard of them. Okay. It's a big trash bag. They'll put everything in there. It can be radioactive. They don't care. Dump. They make a thicker cell wall. They really don't mind, it turns out. How well do cows do if you make them radioactive? Yeah. Hulk cow. Cow angry. Cow make green milk. I don't like this man. All right. And obviously, this has a direct application to biomedical research. Anything we can figure out in terms of protein control in plants, we can then potentially tie back into a biomedical term. And there is money for that. It's not near the money for other diseases that actually kill us, but there is money available in that. There is funding, there is an interest, there's need. Okay? All right. I have also diddled and doodled with the idea of making food more healthy. You may like spinach. Dr. Councilman and I spent 5 lovely years working with spinach in the dark for hours. And so when I see spinach or smell spinach or even hear the word spinach, I get that little lump in the back of my neck that just does this I just kind of do this the rest of the day. Stretch back out. Spinach has a zing to it. That zing comes from calcium oxalate crystals. Here's a problem with spinach. We tell women to eat spinach because it's high in iron. Here's the problem, ladies. If you eat spinach, which is high in iron, you're going to deplete your calcium level because the oxalate will solubilize in your stomach from the calcium oxalate, and it will grab all the calcium in you, which will then lead to bad, very good, osteoporosis. Hey, wait a minute. So how do I offset that? Drink an 8-ounce glass of milk when you eat spinach. You'll be fine. One of the things I've been asking is, can we make food crops healthier? Can we eliminate or at least decrease oxalate loads? I actually got pretty good feedback on a grant for that a couple times. I didn't get funded, but I got good feedback. Delia played on that project a bit. Delia is now a master's student at University of Florida, Gainesville. Okay? She's working with bugs. She liked working with plants so much she now works with bugs. No, it wasn't quite that bad. We were looking at, can we manipulate plants to not pick up oxalate, to not absorb it from their environment? I have several of those projects going on. So if you like working with the idea of making healthier food, come talk to me. All right, I should acknowledge people who have helped. Dr. Webb, Mary Alice was a wonderful postdoc advisor. Gave me complete freedom. Dangerous option on her part, but complete freedom. And she allowed me to hire the undergrads or find the undergrads. And I was blessed to have many awesome ones as a postdoc. Purdue and Wabash were kind enough, along with my previous institution, to provide help, infrastructure, cash, equipment, so on. I certainly appreciate all those things. I'm getting that here now as well. You met Austin and JP, who are now in medical school. Jessica's a PhD student. Jessica Tartaglione is a PsyD, she's a PsyD candidate at the University of, which one is it? It's one of the Chicago schools. I don't remember which one. Carrie's a medical student. Cheryl's going to be a genetic counselor. Sammy's a teacher. Jessica's going to be a preacher. She's been doing that anyway. She's good at that. Davida, I'm sadly enough don't know what happened to him. And Delia is working on her master's. And again, I had huge classes of people. So I didn't have just individuals. I had classes of all of you working with me. Okay? I need your help. Okay? I can't do all this work on my own. And I have just enough energy and just enough naivete to think I can do all of it. I need your help. Okay? Some of it I'm going to coerce from you from my classes within reason. Others, come talk to me. If you have interest in engaging in research, if you want to figure out why, if you have that question, why, in the back of your head, come talk to me. It'd be great. Now, those in my classes have already learned the joy of how so, why, explain. Jessica used to sit there and tell me, why do you write that on there so much? Because you don't answer the question. You don't explain. You have to explain. It's how I know you have it under control. All right. Gentlemen, let me explain something to you very simple. You will pick the one when God tells you. And when He tells you, grab her and never let her go. Never, ever let her go. She will make you a better person. She will make you a better man. She will force you to be a good husband. She will bring you closer to Christ. But understand, once you know, don't let her get away. Don't stalk. Big difference. No stalking. No stalking. But be appropriately aggressive. Ask her out again. Ask her out a third time. Ask her out a fourth time, because you will never regret it. You will never regret that decision. Because, gentlemen, you are not strong enough to generate that. And that, gentlemen, will teach you patience, kindness, and joy like you would not believe. I was not a sharing individual before my boy. I love raspberries. I love raspberries very much. And I have given that child every raspberry I have had since he was born. And I have never felt guilty about it. I've never been angry about it. Trust God. Ladies, I'm speaking to the men because I am one. It's not that I don't love you and don't respect you. And I appreciate everything you do for us. Don't misunderstand me. I truly do. I don't deserve my wife in any way, shape, or form. But, gentlemen, listen to me very carefully. If you want to find out what not to do, come talk to me. And I will tell you. You'll still make the mistake. Oh, no, you will. Because, yeah, you're smarter than me. Sure you are. I was smarter than people talk to me. Come talk to me. I will be happy to talk about my path to Christ and where I have botched it and where I've gotten it right. or at least I think I have. Okay? I truly appreciate y'all coming today. I know many of you came voluntarily. I do appreciate that. Those who didn't, I still love you. If you have questions, holler.